Hello, welcome to our lecture number 5 on single-cell high-throughput data science. This lecture, we are going to cover clustering analysis and its application in single-cell RNA-seq analysis. Let me remind you the whole workflow of single-cell RNA-seq analysis. So we start from a preparation of single-cell samples, and then you load it up to the 10x genomics Chrome controllers, and this controller will prepare the library for you. And that's the single-cell RNA-seq library, and it's a GMBs. Then you send your library to sequencing, and then you get your sequence back. And then you will use Cell Ranger, which is a software solution provided by 10x Genomics to precise your data, to do the mapping and also the count. The output of this pipeline is the gene expression matrix, which we call the UMI matrix, that contains thousands of genes in this expression level in thousands of cells. Okay, cells, that's genes. So we are going to process this matrix next. So this matrix will be the input of our next step of analysis. Last lecture we mentioned about the dimensionality reduction algorithms. We can use one of them to reduce the dimension so you can visualize the distribution of the cells by their similarity. And this lecture, we are going to introduce how are you going to identify the clusters among these data points. So that's our focus of this lecture, clustering analysis. So this is a basic for cell type identification, because we want to group cells first, then you want to use marker gene information to determine what the cell type of this group of cells are. Last lecture we mentioned about the PCA analysis and also tSNE. tSNE is a preferred algorithm over PCA because we tried on the same data set. The PCA will give you a less descriptive picture, so you cannot separate those cells in a higher resolution. Well, the tSNE algorithm will create output, allow cells to be more dispersed, scattered around the space, so you can see the relationship among highly similar cells. Because this is high dimensional data, when you reduce the dimension to 2 or 3 dimensions, you lost a lot of information, and you still want to keep the global structure, but you want to separate the cell to visualize them. So tSNE is a preferred algorithm. Last time we didn't mention how tSNE is working under the hood. I just mentioned that tSNE give you the effects that spread out the cells into more less realistic but more visually appealing distribution, just like this underground map. So although the real distance between the stations is different after you did this transformation, but it's more useful after the transformation. So now I want to spend a couple of minutes to go under the hood to see how this tSNE algorithm is conducted and what's the algorithm behind it. So tSNE the idea started from this paper published in 2003 by Geoffrey Hinton. So his paper is called the stochastic neighbor embedding. Okay, here he proposed this idea to use a probability to measure the relationship between two objects. In our case, the two objects will be two cells. So you can think about this xi is cell number, cell I, and this xj is a cell j. So use their distance between these two and also put into this equation. So if you remember what's the PDF, the probability distribution function of normal distribution is, and this is basically the normal distribution probability. What that means is if you have a normal distribution, and then that's the middle of the zero here. If the distance between xi and j is this, so this from xi to xj, the distance between them, this is the difference between them, and then you will translate this distance into a probability, which you look at this, and then the value is that. If you have a close distance, these two cells are so similar to each other in their expression profile, then this distance is small, and then that will translate into a high probability. So that's the denominator in here. The denominator is the sum of all cells of data points under the consideration which different from I. So this ratio is a relative probability between this probability against the sum of this probability. So at the end, they use the probability of this probability between I and j, the similarity between data points I to data point j to I is conditional probability. So they use this to measure the difference between two data points. In our case, two cells. But once we have this definition, we are going to map this high dimensional space relationship between data points to a low dimension. So if you consider this as the proportional, the conditional probability between I and j, basically the distance between them in the high dimensional space, and this q represents the relationship in low dimension space. So you define both of them, and then the algorithm will try to create this probability relationship with distance between data points in the lower dimension, and at the same time try to minimize the distribution distance between data points in the high dimensionality and also space and in the low dimensionality using this KL divergence metric. Okay, so this KL divergence measures the difference between two distributions. So we aggregate all this and try to minimize this divergence, so to keep the same distribution distribution relationship between data points between high and low dimensionality space. So that's the idea behind the stochastic neighbor embedding. You compute the neighbor by their conditional probability instead of a direct distance. And then in 2008, Dr. Hayden and his student published this tSNE algorithm. At that time, there's no single cell data available yet. Their algorithm is applied to other domain of the of data science, but still later people borrow the algorithm to the single cell RNA-seq. But the characteristic of the tSNE is tSNE, unlike PCA. PCA is not it's for linear projection. So PCA is the linear projection, but the tSNE is not. It's not a linear projection. It can be used to do this nonlinear structure dimensionality reduction and create probability distribution and use the Gaussian, which is normal distribution, to define the relationship between points in higher dimension. And then the trick here is in the lower dimensionality space. They are not directly use Gaussian distribution. Instead, they use student T distribution to recreate this probability distribution among data points. I will explain why they change this rather than use Gaussian. So after doing this, the algorithm will optimize the embedding and the change of the location randomly, and until they found the best solution, and this is an optimization problem. So now I'm going to explain why in lower dimension space they use T distribution instead of a Gaussian distribution. So remember, we have to map the distance from the real distance, Euclidean distance, to the probability by looking at to transform the distance from the distance to probability density, basically. If you transform this using Gaussian, you get something like this. The T distribution, the case distribution is a special case of a T distribution. So it's still T distribution, but the freedom degree of freedom is 1. So you see, for the same distance, the T distribution will give you more spread between a lot of distance. Okay, if input the distance, it will allow you to more spread out. For the same distance, the Gaussian distribution will transform your original distance to a more closed, more smaller distance between the data points. Well, the T distance will have a much large transformed distance after the transformation. So that's why in the low dimensionality, they choose to use T distribution to represent the transformed distance between data points. Okay, I think this will be a little bit tricky to understand, but let's move on. And I'm going to give you real working example. So here is data generated by ourselves, and this is from the mouse brain, stem brain tissue. We obtained some microglia and astrocytes and some other choroid plexus data cells. So before that, before we can decide the cell types, we have to cluster cells into 3 groups. It's obvious from our this tSNE plot. There are 3 main clusters. However, I will let you know this is not a straightforward. If you use most commonly used clustering algorithm, you may not be able to get the correct answer. The problem is that there are a density difference. You see this is a lot of astrocytes dispersed and with a low density. So not every clustering algorithm can capture this signal and correctly assign them into groups. Well, let's go back. There are 2 types of clustering algorithms. One is hierarchy algorithms to create basically a tree to decompose the set of the object into clusters. And another clustering algorithm is called partitioner algorithms that construct various partitions, and then we can evaluate the effects of partition and obtain the optimized solution. Dendrogram is useful tool, and the people have used it for clustering purpose. So we know you can build a tree, and based on similarity between objects, and then you have a node, you have a root, you have leaves. People have used this tree structure everywhere, and this is an example in the molecular evolution. You can build gene tree and to reflect the evolution relationship between different species. When you apply this to clustering examples, we can build distance matrix first between data points, and then start to build this tree. And you can see you can separate this data point by looking at the internal branch of this, and you have, if you cluster this data point into 2 groups by looking at this level of separation, you will clearly separate the 2 groups of data. And their potential use of this dendrogram is to detect outliers. So this particular data points is so deviated from all other data points, no matter which group of the data points. And in the tree, you can see clearly there a branch has been branched out, and you can use this to identify there's an outlier here. So indeed, people have developed a software package for single-cell RNA-seq analysis using hierarchy clustering algorithm. And this is one of the published tool called DendraSplit. And to use this to cluster cells from a single-cell RNA-seq data. So that's the expression matrix. And then you process it and then compute the distance matrix between individual cells. Then you apply dendrogram and to identify the clustering. And you probably have to merge some of the cluster to finalize the results. The problem with this hierarchy clustering algorithm applied to single cell study is It's computationally intensive. This require long time computation for large number of cells. And also their problem is slightly not so easy to interpret the results. You, for example, you don't know which level you want to cut the tree and then separate the cells into groups. So partitioning algorithm, clustering algorithm is still the most commonly used approach in single-cell RNA-seq study as well. This is a non-hierarchy based, and you create this partitioning and then separate them, your original data into different groups. Mathematically, the problem can be translated as a optimization problem. You want to minimize the within cluster sum of square. Okay, and you try to find the central node of your clusters, and then try to minimize the distance between the central node of your cluster to all the member data points belongs to this cluster. And then you have to optimize this procedure to make this global within the cluster sum of square minimum smaller as possible. So most commonly used partitional clustering algorithm include k-means, db-scan, affinity propagation, and published in this paper, and also density cluster, and also spectrum clustering. So we are going to introduce k-means in more details, and then I will show you the performance of spectrum clustering using real example running on MATLAB. Now I'm going to explain how the k-means algorithms perform. So the first step, number one, is you have to determine a value of k. That's where the k-means come from. The number k is predefined. That's the number of the cluster you want to get out from the data you input. So this cannot be automatic. This is a shortcoming, the problem of the k-means. You have to define the k equals to a number. So for example, number 3, then you will get 3 clusters out of it. If you think your data has 6 clusters, you have to define k equals 6, then you will get 6 clusters out of it. And then once you determine the C, and then the next step is to find initialize the k cluster centers. So first, the algorithm will do it randomly. If there's no prior information is given, then the next step is it will decide the cluster member. So all the data points which close to the center of the k clusters will be grouped into different clusters based on their nearest relationship to the center. So after that, you will re-estimate the k cluster centers. They run at the very beginning, step 2, you randomly put the cluster centers, and now you have a re-estimation of the cluster centers by using the cluster members. And then you repeat this process until there's no membership change. So we call it converge. Yeah, so that in the inside the algorithm, you will compute this within cluster sum of a squared distance as a measure of how good of your clustering algorithm result is. Visually, you want to minimize the distance. So if you think that's the center of the cluster of these two, you want to minimize the distance, the sum of distance of this within group. So that's group 1, this group 2. You want to minimize all this distance sum across all those data points. So that's the data points, and that's the center of the two. And after you finish computing for 1, and then you want to do both of this in this graph k equals 2. So that's your objective function for optimization. Now I'm going to use a numeric example. It's a visual example to show you how exactly this k-means algorithm is performed. So for example, you have this distribution of your data points. You can think about those as cells, and then in the tSNE dimensional reduction embedding, you have the relationship among cells like this. So if using these coordinates as input to give to the k-means algorithm, the first step for k-means is randomly select data points and use them as a central node, at the center of your cluster. Obviously, here we have 3 different centers of the cluster. So in this case, we set k is equal to 3. So that's why the algorithm randomly selects 3 data points, 3 cells as the center of this k. So you have a blue cluster, green cluster, and this orange yellow cluster. This is purely random. So you this is just one instance in k-means algorithm. You will run this a couple times until the results are the same. So this is just one instance of the random assign of central noise of the cluster. So that's original data. So you assign 3 centers, central noise of the cluster. And next, you're going to compute the distance for all the data points to this central node. For example, you compute the distance from this data point to blue central node. You just randomly select it as a center, and that's the distance you compute your clean distance by the x-y axis, the coordinates of the data points. We do this for all data point to this blue. Okay, at the end you will find you will do the same for green as well. So you will compute the distance for all the data point to green, and then you will compute all the data points to yellow central node again. Then you will decide whether this data point is close, which central node is most closely related to each data point. Then you assign the data point to its closest neighbor, nearest neighbor, central node. So in this case, these 3 data points have most close, the nearest central node is blue. So they will be assigned into blue group. Similarly, these 2 will be that. So at the end, we will find the nearest cluster center. Okay, so we found the nearest cluster center for the blue data points. Here, these 2 green, and this whole that belongs orange. So once we finish the first step, we are going to recompute the center of this cluster, each of the cluster. So we recompute the center of this blue cluster and put this hypothetic center in the middle of this the group. This is in the middle of the group of green, and that's in the middle of group of orange. Then we will repeat our first step. We're going to recompute all the distance from a data point to each center. This time we use hypothetic center, so you compute all the distance from them to the green one to them as well, orange one to them. So you do this again. So after compute all the distance again, and then you will let those you will let this data point to find the nearest center again. So for example, these 2 orange, 2 yellow data points in the first round was assigned as a yellow group, and now you recompute its distance to the center, and now they become more close to the green center rather than orange center. So this round, they will be grouped into the green cluster. So let's see. So we recompute the distance to all the centers of the new centers, and then reassign data point to their nearest center. So that's a recluster. And then after you finish that again, you will recompute the center of your cluster. For example, the original, the last step, the center of the green is here between these 2. Now it should move to the new center because you add two more green dots in the newly formed cluster. So that's where the re-established center by the last round of the reassign of the membership. So after finish this, we will repeat the exactly same procedure, recompute the distance. For example, we recompute distance about, for this data, to 3 central nodes. And then this is more close to green rather than orange. So next round, this orange data point will be reassigned into green group rather than orange group. So that's what the next assignment will be. So this procedure will go on and on until there's no change of membership anymore. So this is called a converged solution. So you already reach to the optimization basically, and this already minimizes the within group distance, and the sum of all overall is small. So that's how the k-means algorithm is running. Okay, computationally, this algorithm is relatively computationally efficient. It converges quickly to a so-called local optimum, but still will be acceptable. And the problem with this is if you have a cluster, have a shape of so-called non-convex. Okay, so if you have a shape like this or that, this shape is called a non-convex shape. In that case, the k-means cluster will not be really accurate to group all those data points in one group. This algorithm will perform very well for those convex shapes. Okay, so that's the weakness of this. Indeed, when we look at this k-means algorithm applied to some example data, for example, in the first data set, this one, the real cluster was blue and red data points, and the k-means did a very good job to separate them as we expected. However, if you have this setup of two clusters, but the shape of the cluster is non-convex, like this and that, then the k-means will not be able to do a very good job. The outcome of k-means will like this, is 1 and 2 in the red and blue groups, not as what we expected to see. This is a setup example. Similarly, if you have a very complex global structure among your data, and the simulated data have a 1, 2, 3 clusters with the cells arranged in this nonlinear relationship among cells, and then k-means will not be able to capture those signals, and instead it will give you the results of one group here and one group here and the third group here. So not the very good algorithm to apply to a data with a structure like that. The last example is also you have one of the cluster has this non-convex shape, although this one has a convex shape. So when you apply k-means algorithm give you two clusters, and with a obvious and not a very accurate solution. And a lot of this data points was orange has been classified as a blue. Okay, we have our working example. I'm going to show you in MATLAB and how k-means can produce full resolution of your data. This is generated by k-means for this data. And then if we use spectrum clustering, you can see this will generate correct separation of the three groups. So now I'm going to switch to MATLAB and then to show you the live demonstration. Okay, this is my MATLAB interface. So I'm going to load my example data. So this data contains over 1,000 cells, and I already used tSNE to obtain the lower dimension presentation of the cells. So you can see This is what it looks like. As I already see in the previous slide, we have 3 clusters. Obviously, we can detect them visually. Now I'm going to apply the k-means, and I set k equals 3, say k equals 3. And then y use k equals 3, say k-means. So this k-means algorithm takes the coordinates of the tSNE and set k equals 3 to produce a result like this. The first cluster is indicated in green. So here the second cluster is blue, and the red is the third cluster. So this clustering results is clearly counterintuitive. We don't expect it. So k-means algorithm is not a good solution for this. Of course, all the shapes of those clusters are all convex. However, because of the density difference, there's less dense data points and the confused k-means algorithm. So now we are going to try a different algorithm called the spectrum cluster. So you see the input of this function the same as a k-means. You give the coordinates of the cell in tSNE space, and then allow the spectrum cluster to found 3 clusters. And this time we get very good solution, and that's as we expected to see in the 3 clusters. So based on this assignment, you can start to detect what's the marker gene expressed in this group, and in that group, and in this group. Then you will get the assignment of the cell type, because we have a database for marker genes. We know what gene will highly expressed in astrocytes, and what marker genes will be highly expressed in microglia and other cell types. Once you have this clustering algorithm result and put the cells into groups, you can start to compute those specifically expressed gene and specific to particular group, and use that information to assign cell types. Okay, at the end of this lecture, I'm going to, last slide, I'm going to show you the one of the consensus clustering algorithm for cluster single-cell RNA-seq data. The software, the method is called SC3. It's so far regarded as the most accurate method in the single-cell RNA-seq clustering algorithm. It's not based on the tSNE or PCA or UMAP data points, the structure, because it's computed from the expression matrix directly. So you have this UMI, or gene expression count matrix, the gene and the cells. This algorithm will first filter genes and to enrich those genes, give you more information and gather the lowly expressed gene, for example. And then the next, it create N by N the cell by cell distance matrix. We create 3 distance matrix, one using Euclidean distance, another using Pearson correlation, and also Spearman correlation. So 3 distance matrix, that's distance between cells, using their gene expression profile as a variable to compute the distance. Then the next step of the algorithm is transformation. So they transform each of these distance matrix using PCA transformation and the Laplacian transformation. So each distance matrix was transformed into 2. So now you have a 6 transformed distance matrix. And then they select a range of D. D is the number of the components, because all these generated components, and they rank them. So they choose to use from D1 to D different components to learn from the distance matrix, and then use this as input to run k-means algorithm. You see, from each of these truncated transformed distance matrix as input, you can create clustering using k-means, and you run this across all the range, across all this transformed matrix. At the end, the strategy of the SC3 is to use consensus. Okay, so they superimpose all these results together and combine and get the consensus support of this. Any of 2 cells belongs to the same group. At the end, you get your clusters, and for example, those cells in the one cluster, and then there's another small cluster. That's another, then you have a large cluster here. That's from the consensus of the k-means. So k-means is a key algorithm, but in SC algorithm, not apply k-means directly to the tSNE coordinates. So there's no dimension reduction. Instead, SC3 apply k-means to this transformed distance matrix. And also, it's not only use one set of information, but combined multiple set of information, 2 different transform and a different range of the details of the reconstruction of the original distance matrix to get the consensus solution. So that is the main results, the algorithm, how the algorithm is running. Okay, in the last 10 minutes or so, I'm going to show you how to do matrix multiplication. This is the basic skills in linear algebra, and it will be very useful later in our understanding of those decomposition and also the rotation of the data points, the coordinates. So, for example, we are going to compute a matrix of this, multiplied by a matrix of that. First thing we have to mention that you have to make sure the inner dimension of the 2 matrices should be the same. So the dimension of this matrix is 2 by 3, and the dimension of this matrix is 3 by 2 by 2. So in order to compute the multiplication of two matrices. These two matrices have to have this dimension the same. Okay, the second dimension of the first dimension of the second matrix should be the same. Then you multiply this by that, this position by that, and this position by that, and then you sum them together to get the value. Then you do the same for these three numbers against these three numbers, you get value here, and these three numbers against these three numbers to get value here, and these three numbers and that three numbers get value here. So after the matrix multiplication, these two matrices, and the product of them become a 2 by 2. Okay, the inner product. Okay. So let's see how can we compute the multiplication of these two matrices. This is a matrix of 2, 1, and this is 1, 3. So let's put them in the position of this dimension of 2, 1, 1, 3, and these two are the same dimension, and the end product will be a matrix of 2 by 3. So how can we compute? So we will compute this multiplication. This should be 3, and this will be 4, 5, and then 6, 8, 10. So that's the answer. So we compute the first position, multiplication, and then the second, then that's the answer. So the multiplication of these two, the answer is a 2 by 3 matrix of this. So now we are going to use an example to show what's the meaning of matrix multiplication. So this is a interesting example. So there's a hypothetical scenario. A net, that's yourself and your roommate, the two person, want to make a number of a sandwich of 3 cans. So you have a 3 cans of sandwich, 1, 2, 3. That's A matrix, and this you, and then you roommate. You want to make 4 sandwich of the first type. You want to make 5 sandwich of second type, and then third of this. That's 3, 3, 6 for your roommate. So that's matrix A. Then each sandwich is made by putting together some combinations of slices of bread, peanut butter, jelly, ham, cheese, and tomato. So this B matrix will give you the recipe of this, how to make 3 different sandwich, 1, 2, 3, 3 type of sandwich. And the sandwich 1 required 2 slices of bread and 4 units, 4 ounces of the peanut butter, and then 4 ounces jelly, and no ham whatsoever. In the second sandwich, you have the recipient of this, and third of that. So what's the multiplication, the product of A and B? So this basically tells you how many units, how many slices of bread you have to buy, how many ounces of the peanut butters you have to buy, of all these components, of all these ingredients, in order to make 4, 5, 3 type of sandwich. So that's the meaning of this product of the two matrices. Okay, so let's put this into a multiplication. You have 2 by 3 matrix and the 3 by 6 matrix, and this the results of this. So now you know what's the meaning of this if you consider this as a components. So you have 3 components, and then the different factors to contribute to make the components given by the B matrix. And then you have some amount of these components, and this somehow called loading. And then you want to multiply it by B by A, and then you learn what's the outcome of this combination with 3 internal components in there. Okay, so this is the meaning of the matrix multiplication, and you put components into together and see the outcome of this total. There's opposite operation if you are given a matrix of this, and we know it's a product of 2 matrices. There's a way you can estimate or approximate the matrix of A and B. So this process is called decomposition. Okay, we will use this idea later. Okay, so decomposition is opposite as a matrix multiplication. We decompose everything together, and then this decomposition, and we'll be using the product of the 2 matrices to estimate, to guess what the original A and B matrices are. All right, that's all for this lecture, and see you next time.